Hello, my name is Virginia Adams, and I'm an applied scientist on NVIDIA's conversational AI team. Today I'm going to be presenting my and my team's submission to the Track 1 relation extraction task as part of the BioCreative 7 workshop. First, I want to introduce myself and my team. There's me, Hu Chang, Anas, Bo, and Carol, all of whom worked really hard on our submission for this track. Next, I wanted to talk about our vision and goal that we had on the onset of our work here. So we're asked to do relation extraction in which we are given a gene and a chemical, and we want to predict the relationship between these two entities given a context sentence that contains both of them. Because we are participating in multiple tracks as part of this workshop, we wanted to create a model that could perform more than one information extraction task at once. Specifically, we decided to try prompting as a method of doing relation extraction in addition to the other tasks as part of other challenges, where we take a pre-trained generative language model, specifically T5 and GPT, and we formulate relation extraction as a text-to-text task, where we make relation extraction a question that we ask the model, and then it predicts a text output that contains the specific relationship between the entities in the question that we asked. Before I dive deeper into our text-to-text approach to relation extraction, I do want to go over our baseline approach that we compared our text-to-text approach with, and also that ended up performing better in our final submission. So in our baseline approach to relation extraction, we take a more traditional methodology. We start with a pre-trained encoder model that then has two fully connected layers appended after it and predicts a distribution, a probability distribution over the 14 possible relation classes, where one of the classes is no relationship at all. For pre-processing the data before we feed it into this encoder model, we tried two different methodologies. On the left-hand side, we have the tree LCM format in which we mask every chemical and entity appearing in the sentence in the hopes that we keep the model from overfitting to specific chemicals and genes. And chemicals and genes that are not under investigation, that we don't want to predict the relationship between, we mask with an other mask. And then the specific chemical and gene that we do want to predict the relationship between, we have a special chemical and a special gene mask. On the right-hand side, we have the SEM-eval format, in which we completely ignore every other chemical and gene in the sentence, and then add a tag around the chemical and gene that we want to predict the relationship between. So our pre-trained encoder models, we tried several pre-trained encoder models. We tried just vanilla BERT-based, we tried BioBERT, and we tried BioMegatron. We found that BioMegatron worked the best, and we generally saw the trend that larger models worked better, and models pre-trained on biomedically relevant data also worked better when performing downstream tasks, which is something that has been seen multiple times during the literature. Now let's talk about our text-to-text approach. So this first slide is showing what we initially tried. We tried feeding the model an entire paragraph from the PubMed articles that were given, and then we created this type of grammar in which we pulled out the specific named entities from the article that I have highlighted in yellow, and then we tag them with whether they're a gene or a chemical, and then a specific ID number associated with them that I have highlighted in blue and in pink. This whole thing under context is what we input and we prompt the model with, and what's shown under ground truth is what we expect the model to output. So we expect it to output the chemical and gene IDs of the chemicals that it's referring to, and then the relationship that it predicts between them. We found, though, that such a large grammar and prompting the model with the entire paragraph at a time, we ran into max sequence length issues where both T5 and GPT can only take in maximum 2048 tokens at a time. And so we had to move on to a different format. This is the next thing we tried, where we remove the grammar and we phrase it as a more human readable English question, where we say, find the relations, and then we give it an entire context sentence, and then we hope that it outputs every single relationship within that sentence. At least that's how our ground truth training data was set up. We found that the model only ever predicted one relation at a time, and frequently never predicted none as a relationship, even though that was present in the fine-tuning data set. We then moved on to our most successful text-to-text format, in which we explicitly ask it to ask it if there exists a relationship between a chemical and a specific gene. We give it the entire context and we have it simply predict yes or no. And then if a relationship exists, we say predict that relationship, and the only thing the model has to output is the text description of the relationship between that gene and chemical. Finally, this is our results on our development set. Our baseline approach worked better than our text-to-text approach with a couple of caveats. Our best baseline models were pre-trained on biomedically relevant data, where our text-to-text T5 model was not, and our text-to-text GPT model was not. And also our best models were cased, whereas our text-to-text models were not cased. Even so, our very first baseline model did perform worse than our best text-to-text model, which is exciting and promising. And here are our final results on our test set. Our best model was an ensemble of our baseline models, but we think that the text-to-text approach still shows some promise, and there are still more experiments to be done. Thank you.